Here at the University of Delaware's College of Agricultural and Natural Resources, I've spent my summer doing research on rice, glutathione, and safeners as part of the university's Summer Institute program under Matt Limmer. Heavy metals in products we consume is a current public health concern. One such food is rice, as it has a high uptake of arsenic into its roots, stalks, leaves, and grains. This is because arsenite in soil becomes much more mobile in the flooded rice fields, making it easier to absorb into the roots. It is then able to translocate into the rest of the plant body. The arsenic in the grain is toxic to humans, so developing preventative techniques is essential to public health. One such way a plant naturally detoxifies arsenic from its system is through an antioxidant called glutathione. Unbound glutathione, called GSH, binds to the arsenite and brings it to the plant's vacuole. There, it is stored to effectively remove it from the cell system in the form of GSSG. It is theorized that raising concentrations of glutathione would allow the roots to absorb more arsenite, meaning that less would translocate through the rest of the plant to the edible grains. One way to raise glutathione production in plants is to introduce safeners into their system. Safeners are chemicals that typically protect crops from herbicides by raising the concentration of the plant's antioxidants or by altering its metabolism. For this study, rice plants were exposed to 4 different safeners that enhanced glutathione. After exposure, the concentrations of overall glutathione, GSH, and GSSG were determined using UV-Vis spectrophotometry. Oryza sativa, also known as Lamont rice, was raised in a greenhouse in water baths with their nutrient needs being closely monitored. Once the rice was properly grown, 4 pots had 1 safener each introduced to them, with 1 pot acting as the control. After a week, the plants were harvested, split into their leaves and roots, and frozen in liquid nitrogen. The samples were digested and centrifuged to precipitate unwanted proteins. Reducing and extracting agents were added to isolate the overall glutathione. The leaves and roots also had their GSSG isolated to determine individual GS8 and GSSG concentrations. They were analyzed using the UV-Vis spectrophotometer. Currently, the samples need to be compared to the standard curve and then to each other to draw conclusions. It is hypothesized, however, that the leaves and roots will overall have an increase in glutathione concentration. Samples will have a higher concentration of GSH than GSSG, due to their shelter environment in the greenhouse leaving low to detoxify, so GSSG is not being formed. Hopefully, this research can be applied to reduce our exposure to heavy metals so we can live healthier lives.